Hi, my name is Alice Wittig and I'm a PhD student in bioinformatics working for the Hasso-Plattner Institute and Robert Koch Institute in Germany. Today I would like to talk about a tool we developed called CovRadar. CovRadar is a tool for the molecular surveillance of corona mutations. We focus on the spike protein of the virus, which is the red dyed protein you see here on the surface. We do that because it's a main target also for vaccines and antibodies. CovRadar takes sequences as an input and consists out of an analysis pipeline and a web application. The analysis pipeline is written in Python and SnakeMake, making it scalable and reproducible. And the web application is written in Python, Flask, and Dash with interactive plots and tables you can download and use for further analysis. This is the workflow of the analysis pipeline. We download the sequences and use pBlood to align them to the reference spike gene. As a reference sequence, we use the sequence of the first case in Wuhan. After that, we use ViroAlign to build a multiple sequence alignment. And from that multiple sequence alignment, we can refer the consensus sequences of each calendar week and country, the variants, other statistics, and the phylogenetic tree. You can stop here and use our in-house scripts to analyze the results, or you can decide to upload them to a MySQL database to access them via the app. Here you see a screenshot of the app with filter options on the left side and a PDF-like layout in the middle. This is a consensus sequence table. A consensus sequence is the most common base within the multiple sequence alignment per position. So the columns are the positions. And the first row is the first case, meaning our reference sequence. And the other rows are the consensus sequences for this calendar week. For example, here we see an A to T change in the nucleotide, leading to an N to Y change in the amino acid. Especially this mutation is called Nelly. And Nelly appeared the first time end of last year. Nelly is one of the core mutations in the alpha variant. The numbers represent the coverage in this calendar week or for this position. We can see with that table if a mutation becomes most common. What about the time before when they are emerging? For that, we have the alternative allele frequency plot. Each box in this plot represents a nucleotide position in the spike. And the highlighted box is the RBD domain, short for receptor binding domain. This is the area which is connected to the human host cell. The color represents the frequency of the alleles which are not the reference base, so the alternative alleles. With that, we can see upcoming mutations. For example, here we have the Nelly mutation. The other annotated mutations are also core mutations for the alpha variant. So with this plot, we see something is happening in a certain position, but we cannot see which mutation is responsible for. That's why we have mutation distribution plots for the nucleotides and amino acids over the time for a specific position and a specific country. So for example, here you see the position of Nelly of the United Kingdom. And you can see starting end of November, the rise of the nucleotide T, which also leads to the amino acid change Y. It can also be that nucleotide change does not lead to amino acid change. In this case, we are talking about silent mutations. For the German sequences, we have the information about the primary sequencing laboratory, meaning the laboratory sequencing the positive PCR samples. For the laboratories, we also have the postal codes That's why we can't provide these maps. If you have the postal code information for your sequences, you can also expand the map to other countries. I would like to end with a short summary. So CovRadar is a tool for the molecular surveillance of the corona spike protein, and it consists out of an analysis pipeline and an interactive web application you can find on covradar.net. Thank you for your attention. If you have questions, feel free to write them on the poster page. Bye.